So we are going to talk a little bit more in detail about urine formation, and to talk about urine formation, we need to talk about the blood flow through the kidney, because it is all of this stuff that gets filtered or secreted out by the kidneys that will ultimately become urine and will be excreted from our body as waste. And so we talked about functions, overall functions of the kidneys and their responsibilities last week. And so let's just talk about blood flow. So here, this is this region where everything is entering and exiting the kidney. This is the hilum, and entering right here, entering into the kidneys, this is our renal artery. This is bringing oxygenated blood directly from the abdominal aorta. And so this renal artery comes, and then it splits off into arteries that will go up through the renal columns. And these arteries found running through the renal columns, these are called interlobar arteries. And so you can think of it as these pyramids being like lobes, and this interlobar artery is in between the lobes. So interlobar artery. And then when this branches off again and you see the arteries coming around, and they kind of arc around the pyramids, and they are found smack dab in the center, in the middle between the pyramid, the renal medulla, and the renal cortex. This is the arcuate artery. So these are arcuate arteries. So here would be an individual. This would be like one pyramid and the cortex directly above it. So this artery right here coming up would be in the renal column. This is our interlobar artery, and then it branches into the arcuate arteries. Now, when these arcuate arteries branch off again and they give rise to these vessels that run perpendicular to the length of the cortex, and they radiate through the cortex, this is the cortical radiate arteries. Okay, so these are the cortical radiate arteries coming from the arcuate arteries. And then you'll see, so this is in the cortex, and let me point out a few things that I can see for sure. We see these renal corpuscles. Inside would be the glomerulus. So these are renal corpuscles. This would be our of this particular renal corpuscle, this would be our proximal convoluted tubule, descending loop of Henle, ascending loop of Henle, proximal convoluted tubule, and collecting duct. You can see that multiple distals would enter into a single collecting duct. So a collecting duct doesn't belong to any one nephron, but rather several nephrons will empty into a single collecting duct. You can see that this nephron, this would be a cortical nephron, because majority of it is found in the cortex. Only a small portion of the loop of Henle dips a little bit down into the medulla, into the renal pyramids, whereas this nephron, the loop of Henle, dips very far down, and so this would be a juxtamedullary nephron over here. So from the cortical radiate artery, that branches into an arteriole that enters into the renal corpuscle, and so from the cortical radiate artery would be the afferent arteriole, which is bringing blood into the capillary bed called the glomerulus. And so I know that this is the afferent arteriole, one, in larger diameter, but two, it also, you can see these enlarged smooth muscle cells. These are the juxtaglomerular cells of the juxtaglomerular apparatus. So two key things here that tells you this is afferent, not efferent, larger diameter juxtaglomerular cells. So blood comes in through the afferent arteriole, circulates through the glomerular capillary bed. What you can see here, this would be the half without podocytes, and you can see the fenestrae, the holes. So this would be just the fenestrated capillaries of the glomerulus, whereas this half shows you with the podocytes that form that visceral layer, and it's those little pedicels that branch off and form this like woven matrix. That's how we get our slit pores. And so the fenestrae and the slit pores will be our size exclusion. So only the things that can fit through the fenestrae and then the slit pores will be allowed to enter as a filtrate and will be collected in this Bowman's space. Okay? So glomerular capillaries, and then blood has to leave. Blood is going to leave, right? And this blood will be pretty much no red blood cells, no white blood cells, no whole cells or no large proteins will be allowed out into the filtrate, just pretty much water and small molecules, small solutes. So everything that did not get filtered out through glomerular filtration would then exit through the efferent arteriole. Okay? And we talked about why an arteriole coming in and an arteriole leaving is unique. We talked about that last week. So then we have to talk about how we get blood out of the kidney. So then blood exits through the efferent arteriole and then is going to enter into one of two capillary beds. If it's the capillary bed up here, which just, think of this, it would be over top of all of the renal tubules over here. This is the peritubular capillaries. The model is just showing you them by themselves off to the side. These are the peritubular capillaries, which wraps around the renal tubule and the cortex. So these are the peritubular capillaries. Or from the efferent arteriole, it would enter into the capillaries that dip way down into the renal pyramids, and these are the vasa recta. And so just imagine these would be on top of or surrounding the loops of Henle. Okay, so from the efferent arteriole, it's going to either enter into the peritubular capillaries up here or the vasa recta down here. From the peritubular capillaries or the vasa recta, the blood is then going to enter into, you can see it on this side right here. There'd be a cortical radiate vein, and the way out is pretty much the names correspond to the arteries where they're located. So this would be the cortical radiate vein. That would dump into an arcuate vein, which would then dump into an interlobar vein, which would then dump into the renal vein, which then dumps into the inferior vena cava, which would go back to the heart through the right side.